Greetings, everyone. I'm Ben Grumbles, Secretary of the Maryland Department of the Environment, and I am so pleased and honored to welcome you. Thanks for tuning in. We have a delightful and robust discussion in store for you. We have as a tremendous guest speaker, Rita Colwell, and also the one and only Treasurer Nancy Kopp. And just as importantly, we have several young and talented scientists at Maryland Department of the Environment who are going to help engage with Assistant Secretary Suzanne Dorsey throughout the discussion and the presentation by Rita Colwell. But it is my distinct honor and pleasure to also introduce one of Maryland's best known and loved public servants, and that would be Treasurer Nancy Kopp. Nancy Kopp has been elected to serve in the General Assembly. She has served as treasurer over the years. She is the 23rd Maryland state treasurer since the adoption of the Constitution of 1851. She is the second woman ever to serve as state treasurer and the only woman serving Maryland in a constitutional office. She is so engaged in so many important endeavors, public and nonprofit and charitable, serves on the Climate Change Commission, extremely well respected, and I do believe she knows Rita Colwell, our featured speaker, quite well. So let me turn the microphone, so to speak, over to Treasurer Nancy Kopp. Treasurer?
Thank you, Ben. It's a pleasure to be here, an honor to be invited, and a pleasure to be anywhere where I can join you hearing Rita Colwell's latest news. Rita, I think Ben and Suzanne will agree with me. Rita's a force of nature, a brilliant and good woman. And I don't plug people or things very often, but let me just start out by saying, if there's anyone here who hasn't seen this book, I think you can see it. Her latest of 19 or 20 books, A Lab of One's Own, which tells the story that is the story of many of the people in this room and their predecessors, of women starting out in science at a time when academic science and industrial science and even governmental science was not working in their favor. But they made it work. And this book and my 40 plus years of experience with Rita tells me why. Women, including the women in this room, have the capacity to multitask. Rita, in fact, is a polymath, but we can all multitask, and we can all look for the strengths in other people and not be afraid to ask for advice, and not even to be too annoyed when you know that you know what ought to be done, and so you do it by getting a man to say the same thing you just said. I hope that is not as necessary as it used to be, but now that we're getting a critical mass of women in science as in finance, it is important, even more important for the younger women here, as well as the old birds like me, to realize that you too can be role models, you too can be mentors, and the strength of getting things done and getting it done correctly is working together. Rita is a prime example. Rita is a great scientist, and you're going to learn some of the science that she's doing, but she is also a great organizer, a great administrator, a great leader of women and men. She knows how to get things done, how to set priorities, how to establish institutions. After all, she started our Sea Grant program in Maryland, and she created the Maryland Biotechnology Institute, which unfortunately did not last as well as it should have when she became the head of the NSF and the AAAS and any number of other things, while she was writing the 19 books and collecting the 60 plus honorary degrees and having a massif in Antarctica named after her. I'll never forget the day my husband came to me and said, "We just got a postcard from Rita from Antarctica." She must have had a little time on her hands. Rita is someone who knows and has learned over the years, knows how to reach out and bring people together, to be open and to understand that you have to work with people as individuals, not as labels. She's done that, and we should all do that. And this world would be much better if we all did that. These are some of the reasons that I was so overjoyed when Ben said, "By the way, do you know Rita Colwell? Would you mind saying a few things about her?" The other thing I will tell you is that when she came back to Maryland, to academia, she's the only person I know or have ever heard of, though there may be others, who is a named professor at both the University of Maryland and Johns Hopkins University. Usually the two don't talk, much less share the same brilliance. But she has done, as you know, the science. She's done the leadership. She's done the NGO. She's done government at both the federal and state level and the private sector. And in fact, I was pleased, I have to say, to see at the Board of Public Works a month or two ago that you all are contracting with Rita and her company on the project that I hope she's going to mention in her talk, and going to be saving even more lives. She, of course, has saved untold lives from cholera. She has brought together medicine, science, ecology, the environment, and teaching, and never has lost sight of the fact that numbers have faces, that people have faces, that we're dealing with real people. And I will end just with a thanks to Rita. She knows I had one young person, I had two children, but one who was very interested in science. I have no idea what, he probably would have become a lawyer like his father and grandfather, but instead he's working in climate change at Rutgers, basically because he met Rita and he learned that she could be a role model for him too. And everything that he has done, I believe, through Chicago, Caltech, AAAS, AGU, it's Rita's fingerprints are all over it. And for that, I will never be able to thank you enough. Rita Colwell, join me in joining the Rita Colwell fan club. Over to you.
Thank you so much, Nancy. I have to say that Nancy was a mentor. She guided me through the vicissitudes of the legislature and taught me so much. So thank you. And Ben, thank you so much as well. The Department of Environment, as I will point out in my talk, played a major role in the work that I was able to do. What I'd like to do today is to share with you science, some exciting science, and also to show how it really can be used in a wonderful way to help people. So I'm going to talk about climate, the oceans, human health, and interestingly, how cholera, may I have the next slide? How cholera, which is a major part of water-related diseases, water-related diseases are really essentially a pandemic. We are in two pandemics. People don't know that. We're in the cholera pandemic, which started in the 1960s, the seventh pandemic. The waves of cholera seem to affect countries, and currently about 50 countries with about 7 million people affected by cholera. May I have the next slide? Just sort of go through quickly here. The Bengal Delta, Bangladesh and India, have been unfairly criticized as being the homeland of cholera. But 100 years ago, we had cholera in the United States, in Washington, D.C., in the Gulf of Mexico, in Canada, in England, and in Europe, as well as Asia. And the discovery that we made, unfortunately, I didn't get that turned off. The discovery that we made was that the bacterium is naturally occurring. and that we can never eradicate cholera because the bacterium carries out important functions, uh, carbon and nitrogen cycling. We have the next slide. The work that we did really began in Maryland. And here is where I'd like to thank the Department of the Environment, because ship time, access to boats to be able to sample the inlets along the Chesapeake Bay was made possible through the Department of Environment and the terrific people with whom we worked. The numbers indicate the stations that even today are still being sampled on a regular basis. But next slide. What we learned was that the bacterium Vibrio cholerae is present in the Chesapeake Bay, but it's present in the coastline along North Carolina, in the Gulf of Mexico. And occasionally we do have a case, maybe one every few years, of cholera, predominantly in the Gulf, where people will take oysters or crabs from banned areas. And here is the host, the copepod, and this is a gravid female ready to expel eggs into the water column. The cycle of the copepod is to release eggs into the water column, but the bacteria play a role in breaking down the egg sac and releasing the eggs into the water column. We developed this very simple model whereby in the early spring months, when the temperature is very warm and the the um, uh, phytoplankton become dominant, the zooplankton feed on the phytoplankton, they become dominant, and then when the zooplankton run out of their food, this then releases the bacteria into the water column. And for those areas of the world that don't have safe water, this is how the bacterium is transmitted. Now the next slide. Now, one of the things that occurred to me, this was 25 years ago, was the fact that there was this relationship to chlorophyll, this relationship to warmer temperatures. And in 1985, a satellite was launched, Landsat. NASA launched the satellite that had sensors that measured chlorophyll, sea surface temperature, and sea surface height. Well, it occurred to me that this incidence of cholera in Bangladesh, which was spring and then fall peaks, was related to the plankton blooms, that we could actually monitor and predict cholera using satellite sensing. And so that's exactly what we did. We then, next slide, showed that there was a real correlation um, between the chlorophyll peaks and the measurement by satellite of sea surface temperature and height. Now here, because we had been criticized, being a woman scientist, if I'd been a man, I'm sure the discovery we made that the bacterium was naturally occurring would have been promulgated everywhere as a new finding. Nevertheless, because what we had discovered in Maryland was disputed by the medical community, some in the medical community, that this wasn't what happened in Bangladesh. Well, that challenged us to go to Bangladesh, which we started our work back in 1975. And indeed, we found that, yes, there were copepods in the water. Yes, that in fact the bacteria were associated with the copepods, also a little bit with some of the other components. But next slide. Clearly, when there's very poor sanitation, next slide, person-to-person transmission is very important. However, next slide, it's really the environment that's the source of the bacteria. Next slide. So now this allowed us this putting together chlorophyll, warm temperature, sea surface height, satellite sensing, and the correlation was dramatic. We published this 21 years ago. This is the first satellite prediction of an infectious disease. And here you see the red line are the actual numbers of cases of cholera in the villages along the coast of the Bay of Bengal. And the blue line the temperature of the water in a 1-mile quadrant just offshore in the Bay of Bengal, which was the sensitivity at the time in 1985 and 1990 of the satellite sensors. So this then showed us that we could use satellite sensing as an indicator of the conditions that would be optimum for the outbreaks of cholera. We published this in the Proceedings of the National Academy of Sciences in 2000, 21 years ago. The results were dramatic. By satellite, we could predict, just show the next slides. We could predict in Calcutta and in Bangladesh, MATLAB Bangladesh, that with the milligram per cubic meter increase in chlorophyll, there would be a 33% increase in cholera cases in Calcutta. With a millimeter per day increase in rainfall, there would be about a 7% increase in cholera cases. And similarly, in Bangladesh, in MATLAB, the research village where we did most of our work. May I have the next slide, please? Now, we've produced a much more sensitive model year by year, and now we have a highly sensitive model which relates the temperature in a preceding 3 or 4 weeks coupled with a heavy rainfall in an area of the world where there's poor sanitation, lack of access to safe water, and then you would have a high cholera risk. Next slide. So we now use much more sophisticated satellites. From 1996, as you can see in the lower left, that was our first foray into satellite sensing and measuring and predicting disease. And now we use JSON and Discover satellite sensors that allow us to measure a square meter of water temperature and chlorophyll, which means we can measure the temperature and the chlorophyll in a pond rather than in a quadrant mile offshore of the Bay of Bengal. In addition, with these sensitive satellites, we can measure the movement of populations, the migration, let's say during strife, during a civil war, when people flee from areas and the other areas become congested. May I have the next slide, please? So this allowed us, next slide, to make the kinds of sensitive predictions. Why don't you go back to the previous slide? I think that might be helpful a bit if you can. Because and if you, well, no, that's okay. This is fine. Leave it. We were interested in the Hurricane Matthew, which traversed Haiti, where the worst cholera outbreak in 50 years had occurred in 2010, right after the earthquake. And Matthew followed 5 years later, a hurricane that came up the coast of the United States as well. We retrospectively analyzed the data. We were able to make a prediction of cholera shown on the left, the actual cases shown on the right. So the model now is highly sensitive. May I have the next slide? We did a similar analysis of Yemen. As you may know, Yemen has suffered the worst cholera epidemic in perhaps ever, but at least in the last 50 or 100 years. It is due to civil strife. There's a civil war, and thousands of cases and thousands of deaths as well. So we analyzed retrospectively in Haiti, and we were able to show, as you can see in the upper right, the prediction map, and the lower right, the actual numbers of cases where they occurred. This was picked up by the British Aid Agency and a phone call in 2018 from the British Aid Agency leader asked us if we would collaborate with them, provide them with risk maps every month that would predict a month ahead where the risk of cholera would be highest. They would locate physicians, medical supplies, safe drinking water supplies, and we would work together. And we've been doing that ever since. So for the last 3 years, We now provide every 2 weeks a map of a month ahead, and we collaborate with UNICEF, the British Aid Agency, NASA, and the British Meteorological Agency for providing these risk maps and for this work to be done. May I have the next slide, please? Now, what I'd like to do is turn to another kind of work that we've been doing on DNA sequencing. Next slide, please. What we do is this was after I had finished at NSF. I knew the country needed a very rapid identification technique because having run the anthrax investigation as an advisor, chair of an advisory committee to the CIA and the FBI, it took us 6 years to make the identification 21 years ago. So what we developed was this technique of extracting DNA from water samples, clinical samples, blood, sputum, um, food, didn't matter. Extracted DNA through any kind of sequencer, matching it up against a database, which we have developed, 160,000 curated, clean DNA sequences of bacteria, viruses, fungus, and protists, parasites. This gives us not only the identification, but also the genes present that code for antibiotic resistance, for pathogenicity, for metabolic properties. Next slide. We can identify right down to strain. Now, this is important because if you're a physician, E. coli is present in our gut, but the strain H70157 causes severe disease. Similarly, Lactobacillus casei is a very good bacterium, but one strain is used to make yogurt, another strain is used to make cheese, and a third strain is used specifically to make wine. So strain matters. May I have the next slide, please? Now, let me give you an example of how we've applied this to water safety. Next slide. In California, they've had a very severe drought, which we all read about, followed by forest fires. Now, we joined up with the Orange County Water District, which provides safe drinking water for the Los Angeles and surroundings. Now, the drinking water plant of the Orange County District was running out of groundwater to purify, to distribute to the citizens of the Los Angeles area. And they were using sewage water, 50% coming in from the sewage plant, wastewater, recycling. We showed that, yes, indeed, in the Q1, the water coming in from the sewage plant is that gray area, all kinds of microorganisms. In this case, I'm showing the bacteria, including some pathogens. But by the time it's gone through the microfiltration and reverse osmosis, the only bacteria are the naturally occurring water bacteria that you would find in the drinking water that you take from the tap now. Next slide, please. Similarly, for viruses, in the Q1 water coming from the sewage treatment plant, the wastewater, there are human viruses, agriculturally important viruses like the pepper mild motile virus. But by the time it's gone through filtration, reverse osmosis, and certainly by the time it's chlorinated, the only viruses are bacterial viruses, and those are harmless for humans. Next slide, please. The concern was that antibiotic resistant genes might be transferred from the sewage bacteria to the naturally occurring water bacteria, but we found that that was not the case, that the only antibiotic resistance came in with the sewage bacteria. Now let's look at COVID-19. You're going to wonder, what is the relationship here? Because this is a virus that causes the COVID-19 disease. It's a virus that attaches to receptor sites, the ACE2 receptor, which is widely distributed on the lungs, liver, kidneys, the intestines, on the heart. This is a surface receptor that seems to be broadly distributed amongst the organs of the human body. It even affects the neural system because we know that one of the dramatic symptoms for severe cases is the loss of taste and the sense of smell. May I have the next slide, please? we determined very quickly back in February and March of 2020, just when the virus was becoming recognized as a pandemic causing virus, that we could identify the virus, the SARS-CoV-2, as well as its variants by the technique that we had developed for DNA sequencing and the bioinformatics for identification and characterization. Next slide, please. So now we took advantage of the information published showing that individuals, this was published probably May or June last year, showing that about half of those infected with the virus shed the virus in their stool. May I have the next slide? Now, this next slide looks a little complicated. It's not. Just read from the top to the bottom. These are 41 patients. You read across. You see the red line. That's the time period for that patient that the throat swab, the nasal swab, was positive. The yellow line, the yellow-orange line, shows how long after the nasal swab is negative that the virus continues to be shed in the stool. So this allows a measurement of the presence of the virus in a community. Next slide, please. And here is where we joined forces with the Department of Environment, Department of Health, initially with the Department of Health in Frederick, Maryland. And we sampled sewage plants. May I have the next slide, please? And this allowed us way back in June and July for Frederick, Maryland. You can see the blue line. That is the detection of the N genes of the COVID SARS-2 virus, the causative agent of COVID-19. You also see the orange line, and that's because we use, in addition, another set of genes to confirm the identification. So we're very strongly convinced that the detection is accurate and precise with respect to numbers of virus in the samples that we analyze. And you can see the peak, the jump um, uh, that occurred in July of 2020, last year. And this was an early warning for cases that appeared in the community later. Next slide, please. The current work is very extensive testing throughout the state of Maryland, where the viral load, the number of virus, gives us an early warning for an outbreak that's either going to happen or is beginning to happen, or we're in the progress of it. And we're able to actually identify the strain and type it. And then in addition, we're doing some elegant analysis of everything that's in the wastewater with respect to microorganisms. In other words, the viral load is detected by PCR, but we use whole genome analysis, the DNA analysis, to determine the presence of bacteria, viruses, fungus, protists, potential pathogens. May I have the next slide, please? And so this is a highly controlled assay that allows us to have a very precise determination of the presence of the virus in the samples that we collect on a regular basis. And you see the steps that are taken for the analysis. May I have the next slide, please? Now, we target the regions of the N gene, which is the nucleocapsid and the spike of the virus, and that gives us then the identification of the virus in the samples that we're testing. May I have the next slide, please? And so, what is fascinating is that this is a public health tool. With metagenomics, now we can map the pathogens circulating in the community on a regular basis to provide early warning for physicians and hospitals to be on the alert for cases. Bear the next slide. This shows that the bacteria in the Back River and in Salisbury, Maryland, a peak of certain kinds of bacteria in the back river samples over a period of time. And these are the kinds of bacteria present, identified down to species and strain that are present in the sample. An abundance of Acinetobacter and Klebsiella, a peak, as you can see on the left, of those species. A very interesting indication of what's going on in the community, those communities. May I have the next slide, please? And so we can also map antibiotic resistance, an indication of people being treated with antibiotics and the discharge into the sewage system. So we now have what we really should have known some 50 years ago when we did test wastewater for poliovirus to monitor polio epidemics, but we kind of let it go by the by. And now that we have had the COVID-19 pandemic, I think we can use this analysis as a very special public health, community health tool. May I have the next slide, please? And so the strain typing allows us to detect the presence of given strain and then to link it with the community isolates from individuals so that we can actually monitor by virtue of, let's say, a variant appearing in the sewage. We can provide an early warning. And then for the patient sampling, instead of just doing the single PCR for the dominant variant that's been used, you could additionally use another PCR for one of the variants. And by linking these, we can really confirm the presence within the community. May I have the next slide, please? And so here's a case study that we completed with Mount St. Mary's with their permission to present the information in which we will publish. We have done a twice weekly sampling of a dormitory. We picked up the virus in that one dormitory. 221 students were tested in that dormitory. 10 were positive, but 9 didn't even know they had the virus. It could have been a massive spreading event, and the university did not have to be shut down to be tested because those coronavirus positive students were isolated and they would continue to be tested until they went negative. And so the wastewater tests are done twice weekly now, and it's a much more efficient and less expensive and more effective way to monitor. May I have the next slide, please? Now, the other thing that we have done is to take the satellite model for cholera, and we've modified it to use the factors that are important for COVID: humidity, dew point temperature, movement of populations by accessing cell phone data that are used to monitor and track COVID cases. You can see on the left a map we made in April, May 2020, a year ago. That's the risk map. On the right is the actual map of cases. You can see the sensitivity. Next slide, please. A more recent map was produced in January, February of this year, showing the areas of highest risk county by county in the United States. So this provides us then with a prediction map for this disease. May I have the next slide, please? What I'd like to do is close by giving a final example. I've shown you the use of satellite sensing, computational modeling, DNA sequencing, the high technology of informatics. But still, there are people in Bangladesh. Those boats are actually their homes. The water they take is from the environment. But what we found, because we had shown that the copepod is the carrier, the vector of the cholera bacteria, that and it's kind of like the elephant of the microscopic world. It's 250 microns. The bacteria are about 2 microns. But we showed in the laboratory that we could crudely filter out by using sari cloth And shown here is instructing the women after they have used the filter to open it up, rinse it, air dry it, because in the sunlight acts as a disinfectant. The water to the right has been filtered. The water to the left has not. It was very easy to convince the women that filtration is important. And in fact, 5 years after we went back to analyze, we found that even the control villages, which we had not taught to filter, were filtering. But the full study showed we could reduce cholera by 50%. So here is science used in a more simplistic way, but effective to save people's lives. Next slide. Lots of people are involved. Dr. Alam, particularly from the International Center of Diarrheal Diseases Research in Bangladesh, Dr. Balakrishnan Nair and Dr. Ramamurthy, the directors respectively of the National Institute of Cholera and Enteric Diseases in Calcutta, and wonderful students and postdocs at the University of Maryland. Next slide, please. And we also collaborate with folks at Ames, the national NASA, and from other universities in other countries. May I have the next slide, please? And much of this is important to focus on the fact that safe water is a global challenge, a bigger challenge in the future than energy. And it's one that we have to overcome. There are 1 billion people right now who have no access to safe water. Next slide. This is written up in my book, A Lab of One's Own. Next slide. And what I'd like to do now is to take questions. Next slide. Very good. I'd like to take questions from those of you who are in the audience, and to thank you all for attending and for your interest. Thank you.
Thank you, Dr. Colwell. We appreciate so much what you do, and we are so pleased that you have shared your story with us here at MDE. It's been exciting for me to watch the overlap, and like I see Treasurer Kopp, I also invited my daughter to join us. So it really is an honor to host you here at MDE. We have with us today, in addition to an audience of many of our MDE colleagues and friends, several future leaders for the Maryland Department of the Environment. I'd like to introduce them, and then I will allow them to ask several questions. First, I want to introduce Cassie Omo Asaji. She's a natural resource planner in land and materials administration. Cindy Osoto, policy analyst for the climate change program. That's right, wave when I say your name, everyone. Joey Adia, regulatory compliance and engineer in wastewater permits and helping with the Sewer Sentinel initiative. Sarah Albrecht, natural resource planner in the Bay Restoration Fund program, also helping to work in the Sewer Sentinel project. And Melissa Wright, the certification compliance head for the oil control program. So I'm going to start out with Sarah. Sarah, I believe you have a question for Dr. Colwell.
Yes, thank you. Thank you, Dr. Colwell. First off, it seems like you know about just about every topic, so it's hard to choose just one question. But as Dr. Dorsey said, I'm working in the SSI sewer project. My question pertained to that. It looked like you guys had a strong correlation between your N gene and your ORF1AB. What we've found is that in a decent amount of cases, one will be present without the other, and it's been both ways where there might be N gene but no ORF1, or there is ORF1 and no N gene. I was wondering if it was understood yet how those interact with each other and how it's possible that sometimes they are not present.
Well, the virus is a relatively small virus, and it mutates very rapidly because it replicates very rapidly. Hence, mutations are more frequent, and the bulk of the mutations are not necessarily of interest because they don't necessarily increase infectivity or severity of the disease. But a few variants do. And so I think the fact that there may be deletions is something that we need to determine. And the fact that we do whole genome sequencing, we, in addition to the PCR, we actually match up the fragments of the virus, and we can determine that the identification by whole genome sequencing is usually 98 to 99% correlation of the genes present with the reference virus. So it's a much more accurate identification than PCR, because we're actually taking into account and matching up all the fragments, so to speak, to restructure the virus. I'm making it a little oversimplified, but I just want to make the point that with the whole genome sequencing, we are matching the fragments and determining that we've got a good strong percentage of the virus so that we know that we're identifying it. So the rapidity of its replication is coupled to the fact that it will be sloppy in replication and will not be 100% in every single replicated virus.
Thank you.
Do we have another question? Cassie?
Dr. Colwell, you said that the cholera infection is basically from the environment, but it can be transmitted from one individual to another. Can you explain how that can happen and how we can reduce the contamination from individual to individual?
Yes, cholera bacterium is transmitted through contaminated water and food and what we call fomites, that is contaminated utensils, doorknobs, that sort of thing. It is not a respiratory virus. It is an intestinal, it's not even a virus, it's a bacterium. It's an intestinal bacterium. And how it acts is that it attaches to the surface of the intestine, and the toxin that it produces causes a sodium potassium imbalance, and really it's a systemic shock that you go into in the most severe cases that lead to death. And so it's really a physical transmission in person to person. I think Nancy Kopp has been muted. Can she be unmuted?
We can't hear you.
I'm muted.
We've got you.
We
Sorry.
do not want a muted treasurer,
Never.
ever.
Treasurer Cop, did you have a question?
No,
Okay.
it's,
Okay.
we were
I know-
discussing, we were discussing it.
Melissa, you have a question.
Yeah, my question is kind of related to your book. I haven't got a chance to read it, so I was glad to be informed on it because I do plan on reading it, but it's related to the sexism in science. And I was wondering if you had any advice for, or any sort of self-advocacy advice for women in the sciences when they do have to face those challenges, either on a daily or not daily basis, but just as a way to kind of keep our heads up or kind of figure out what to do.
Well, the advice I give regularly is get yourself a posse.
Mm-hmm.
What do I mean by that? Meet on a regular basis with other women who are pursuing science or engineering or medicine or working in your area, whether it's science or not, and meet get together for coffee, and every couple of weeks or every month or so, or dinner or lunch or whatever, and talk about the obstructions, the insinuations, the slanders, whatever has come up, and you'll find that you're not alone. And there's a kind of reassurance that it's not you, it's the system. And when you recognize that, then you are able to build your own defenses against it. And I think that's the most powerful advice that I could give. And I think that the notion of a posse has caught on.
Love it. Thank you. Thank you, Dr. Colwell. Rebecca, you had a question.
Thank you, Dr. Colwell, again for taking time today to share portions of your very robust career. I had a question about if you could talk about more how you determined that the copeped's life cycle was the vector that's causing this naturally occurring bacteria of cholera.
This is a very long study that goes back to the 60s. When I first came to Maryland, I started at Georgetown University, and I started collecting samples courtesy of the Department of Environment, um, of oysters and crabs and so forth, having been trained as a marine microbiologist at the University of Washington. And I found that Vibrios were the dominant microorganisms that were present in the coastal areas. They were kind of like weeds. You could pick them up easily. Um, and then I had a student, Dr. Tetsuo Kaneko. Tetsuo came to work with me from Japan because he'd read about the work that I'd done on Vibrios in the Chesapeake Bay. And he did his PhD at Georgetown showing the relationship of zooplankton and Vibrios. And then it led from there to the work of Jim Caper, who is now vice dean of the medical school and chair of the micro department in Baltimore. And Jim did a fantastic study showing the actual presence of Vibrio cholerae and the relationship, seasonal relationship, and salinity relationship, sort of mid-bay. And we're currently doing work on the Vibrios in the Bay, funded by the National Science Foundation, and Dr. Anwar Haq is the PI, and I'm a co-PI with him. And we're looking at Vibrio vulnificus, which is another bacterium of serious concern because it has a 50% mortality associated with it. And victims, if infected, um, can lose a limb or their life. And so we're trying to develop and succeeding to develop a satellite prediction for the intensity of Vibrio vulnificus so that we can provide an early warning to watermen and to recreational folks to be careful. And if they get an infection, treat it seriously. Don't just douse it with mercurochrome, but get yourself to an outpatient a hospital or to a physician to get an antibiotic. So it was about a 30-year study, and we published the work predominantly in the 20 years ago and 10 years ago. And the reason I have so many publications is because we couldn't convince because it was a woman that had made this discovery and it wasn't being accepted. So we just published and published and published. And now there are probably a couple thousand publications, not just mine, but other scientists in other countries have found the same. And one of my students actually isolated Vibrio cholerae from Iceland. They've never had cholera in Iceland. But in the geothermal warm waters, the student was able to isolate the bacterium.
Thank you, Dr. Colwell. I believe Joey has a question next.
Yeah, so this wastewater sampling was used back since polio, and now COVID. Do you see this as a new normal thing for future viruses?
I do. I think that this is a tool, particularly with whole genome sequencing, next generation sequencing, because the work we're doing with Suzanne Dorsey and with Secretary Grumbles and the team at the Department of Environment is an analysis of everything present, bacteria, viruses, fungus, parasites, in the sewage water that allows us to determine a, how shall I say, a pattern for the communities around the state. And it's an early warning system. perhaps for outbreak of Giardia or Cryptosporidium, we would be able to get it quickly and nail it. And working with the Department of Environment, we can confirm the presence that we pick up of a pathogen early on of a pathogen. Then if there's an outbreak of an infection that's kind of hard to diagnose, this is information that allows funneling and focusing the identification to the specific microorganism, bacteria, virus, fungus, or protist. So I think this is a public health tool, and I think it's beginning to be understood because I'm reading papers now. In fact, Johns Hopkins in his alumni bulletin, had a very brief article about the potential of sewage testing as a public health tool.
Thank you. I will just add to that I know MDH is seriously looking into continuing and extending this beyond COVID. So I think the public health sector has begun to realize again to use this resource. Cindy, you had a question.
I come from the world of economics and environmental sciences and policy, so I've been influenced by the interdisciplinary field, and I think one of the most inspiring things about your work is how you have been able to tie in all these different fields to really make a real influence. I'm wondering if you see that extending to climate change in the future as well. And how you think that will progress over time, the interconnection of different fields?
That's a great question, Cindy, because we actually have published a paper 3 or 4 years ago in collaboration with a team in Genoa, Italy, Dr. Carla Pruzzo, a wonderful scientist and colleague. And also this paper was collaborative with the Max Planck Institute in Germany, where they do very elegant DNA work, and the Marine Lab in England, and us at Maryland. And what had been done was the Marine Lab in England 45 years ago had brilliantly developed a citizen science program where they sent plankton nets to yachtsmen, shipping boat captains, cruising boat captains, ferry boat captains, and said, please collect a plankton sample, give us the latitude and longitude, send it to us, and we'll archive it. So what we did was to go to the samples from 40 years ago, 30 years ago, 20 years ago, at different locations in the North Sea and off the North Atlantic coast of the United States, and we extracted the DNA from the plankton samples and then probed with specific probes for Vibrio cholerae, Vibrio perihemolyticus, Vibrio vulnificus, and Vibrio alginolyticus. And we were able to show that as the temperature of the ocean surface is warming, measured by the Marine Lab, that we could see increase in the numbers of Vibrios, pathogenic species, because we were using PCR probes, and then we could link this, correlate it with increased numbers of infections occurring in Europe and in the north of the US and in Canada. And we published that in the Proceedings of the National Academy of Sciences a few years ago. And I think it's the first demonstration documented of an infectious disease actually influenced directly by climate change, namely sea surface warming.
Dr. Colwell, I have a follow-up question from the audience, and I apologize. I, for some reason, can't see the name of this person who asked it, but this is somebody who works in our air department, and she recognizes that she uses modeling a lot with air quality and wanted to know if there were other examples where we've been able to use satellite modeling to track waterborne diseases.
There's work being done on malaria because it's a vector-borne disease as well. In the case of cholera, the vector swims. In the case of malaria, it flies. And I think the notion of satellite as a public health tool has caught on. And there are a number of laboratories around the world that are now joining the bandwagon, and I'm delighted because I think satellites are an important public health tool, and especially the very sophisticated ones that allow measurement of very precise characteristics, turbidity, and even salinity now, which I think is marvelous because salinity is another big factor in several of these pathogens.
Great, thank you. And that was Mola Saros from our Air and Radiation Administration. Kate informs me that we have a question from an audience member. Kate, let's see, James Wagner had a question. James, if you want to unmute yourself and ask your question.
Yes, thank you. Yes, I don't know why I wasn't offered the the opportunity to present a question. It seems the others were. My question concerns any environmental impacts to COVID-19 infection in the vein of the Vibrio. In other words, early on, we heard about possible low incidence of the virus in warm locales. And I'm wondering if there is any data on that, heat, humidity, sunlight, for instance, affecting activity.
That's a beautiful question. And we're investigating it very intensively. In fact, the question about modeling aerosols, that is something that we're very keenly interested in. In fact, my hypothesis is that the virus in microdroplets transports itself across miles, not just meters. And so the evidence is that the relationship has a lot to do with humidity, warm temperatures, and the movement of populations. It's not just a warm temperature, but it's the fact that when it's very hot, you have people move into closed quarters because it's cooler with air conditioning. And similarly, when it's very cold, the ambient inside air is really very important. So the difference between ambient outside and inside air, that's another factor that has to be brought into consideration in this fairly complicated model for the transmission of the COVID-19 virus. But we've been working, or at least beginning discussions with Dr. Kim Prather at the University of California, San Diego, Scripps Institution. She's been doing some beautiful work on airborne transmission of chemicals and particulates and viruses. And she has shown that sea spray, where there's an outlet, will carry and transmit the virus, which is one of the reasons that in California, surfing was banned from certain beaches where the outlet from wastewater was too close to the beach. So that's another very interesting aspect of this virus and its capacity to transmit in microdroplets. Great question. Thank you.
Thank you.
Dr. Colwell, as we come to the close, I want to just ask our panelists if anybody has any questions.
Yes.
I want to say hello to the other important cop family member, Nancy's husband. Hi. Great to see you.
From our panelists?
I have a question, Suzanne, if we have a minute, talking about measuring things different ways and putting your biotech stuff together and climate change. One of the things we are taught is that in 20 years, we are going to be like Mississippi. This is a vague, but But if that were so, how could we use that to learn what to do in Maryland now?
I think it's very serious question. It's a real question.
Yeah.
I think climate deniers are fewer and fewer in Miami, for example, because with every very high tide, which I guess occurs every several weeks, every month, they're out waiting on Main Street. So sea level rise is real, and it seems that it's more that we've been conservative as scientists rather than totally realistic. Well, I guess we're realistic, but we've weighed on the lower side than the higher side. So I think what we need to do is understand that in climates that we are progressively moving toward, such as in Singapore or Vietnam, we should be thinking about Maryland. I certainly think that Maryland, as a matter of policy, should be revisiting or visiting, if it hasn't, policies associated with rebuilding on the shore. Um, Ocean City, these are economic problems, but fascinatingly, the insurance companies are paying attention. And it seems to me that as a matter of policy for the state should be paying attention. So it would not be outlandish to actually have a climate change committee or commission just begin to investigate with the changes that can be anticipated, moving initially toward a warmer climate such as Georgia, and then a warmer climate such as in Florida, and then a warmer climate such as in the Caribbean, etc., that we can understand what the effects would be and predict the effects in Maryland. And that would be an interesting scientific enterprise at the minimum, but at the most, it would be a wake up and a planning, um, both for mitigating and slowing climate change, but also being prepared for the changes that we will find inevitable. So, good question.
Thank you. I'm sure the secretary will want to follow up with you.
Well, I embrace the opportunities for resilience and adaptation, and also for Suzanne Dorsey, who is really stepping up and coordinating with Department of Natural Resources and all the other governmental organizations, but to lead with science on that, on the leaning forward on resilience. Suzanne?
Thank you. I appreciate that. I know that at MDE, every day I am inspired by the young people around me. I get to work with incredible, motivated, skillful, and determined young people. It is, you know, the thing that fills my cup every day. And I also know that I stand on the shoulders of great women like Dr. Colwell, like Treasurer Kopp. And I just hope that we all find a way to work together, to continue to work together, to recognize the good work of the people around us and to elevate the people around us and find ways, like Dr. Colwell has, to make a real difference in this world. Dr. Colwell, I am so grateful that you shared your time with us today. And on behalf of the staff at MDE, thank you for sharing this time with us.
It's been a pleasure and an honor. Thank you so much. And thank you and good luck to the young people. Go for it. Don't let any impediment keep you down. Just keep going.
And read the book.
Absolutely. I want to for another hour just about anthrax, Dr. Colwell.
Yeah. Yeah.
Thank you.
Excellent. Thank you
right.
so much.
Thank
Thank-
you.
Pleasure.
Bye.
Bye,
Bye-bye.
everybody. Thank you.
Oh,
Thanks.
Bye.
Or maybe not.